So dear students, now we will start the composition of chromosomes. Composition of chromosomes. As we all know that chromosomes, they have the hereditary material DNA present inside them, and another component which is very important in the composition of chromosome is the proteins. Proteins are not only the important component of chromosomes, but they are very important components of the whole living organisms. We have almost everything made up of protein. Our skin is protein, our hair, they are protein, enzymes, hormones, all of them, they are protein in nature. So our muscles, they are also composed of complex proteins. So proteins, they constitute the major portion of our composition, and as well as the composition of the cell. So DNA, basically its function is to synthesize particular proteins depending upon the function that is to be performed by them. And the protein, it is manufactured or it is produced depending upon the specific genetic code for that protein. For example, if the eye color protein is required to be black, then the specific gene will be present which will say the eye colors of that individual will be black. So the amino acids for black color eye protein will be synthesized, and hence the individual will have black eyes. So chromosomes, if we talk about their composition, they have almost 40% DNA. All of the DNA is present inside chromosomes, but still they constitute only the 40% of the whole chromosomal structure, and almost 60% is proteins. Proteins are not only structural, but they are very much supportive when it comes to their function. Also, there is significant amount of RNA. Significant means quite important. Significant amount of RNA, ribonucleic acid. Now, what is RNA? RNA is ribonucleic acid, and it takes instructions from DNA. DNA instructs RNA for the synthesis of particular protein. So RNA, it gathers different amino acids and join them to form the structure of a particular protein. Now, we all know that DNA, it is present throughout the length of chromosome. DNA extends throughout the length of chromosome. So the DNA, we all know that it is a double helical structure, a coiled structure, which is present throughout the entire length of the chromosome. And a typical human chromosome, it consists about 140 million nucleotides of DNA. In a single chromosome, there are 140 million nucleotides. Of DNA, and the amount of information one chromosome contains, it will fill about 280 printed books if each book has 1000 pages. So it is a lot of information to be present inside a single chromosome. And if you remove DNA from the chromosome, DNA will be visible using the naked eye because its length is in millimeters. While chromosomes, they are present in such smaller size that they are not visible through naked eye. So the presence of a very large molecule in a very small space is indeed a very big marvel of nature, and that is only one of 46 chromosomes. If you multiply this huge information with 46, you can imagine yourself how much information is allowed to fit into a smaller space. Now the question arises that how a very big macromolecule of DNA is properly fit inside the chromosome. So the proper fitting of DNA inside the chromosome is due to the coiling of DNA. Means like this double helical structure is coiled and it is fixed inside the whole length of the chromatid. Now, how the DNA is properly coiled? The DNA is coiled such that after every 200 nucleotides, the DNA is coiled. DNA is coiled around 8 histone proteins. So the point where DNA is coiled around the 8 histone protein forms a complex which is known as nucleosome. So this is known as nucleosome. The point where DNA is coiled around the 8 histone proteins is known as nucleosome. DNA is negatively charged because it has phosphate group present inside it. So this coiling is facilitated because of that negative charge on DNA. DNA has negative charge and histones, they are positively charged. So this positive and negative charge, they attract each other and allow a very tight coiling. Then there is first coiling, then further coiling, and then supercoiling. So the very good and fit coiling will allow the proper arrangement or fitting of DNA molecule inside the chromosome. So what is chromatin? The chromatin material